So I have a PSA, a public service announcement, as I am getting these slides up here. Some awesome person here who is a Star Trek fan left their keys outside. So they're somebody who drives a Ford van and is also a huge fan of Star Trek with a Star Trek keychain. You can go find Jake, who's back there in the black shirt. Jake right there will reunite you with your vehicle if you go find him. So let's see, I'll pull up these slides. Okay, well, oh yes, here we are, perfect. Thank you so much. This is such an honor to be here. Thank you, Judy, for that incredibly nice introduction. And this is just, well, I'm going to talk about this idea of contending with the truly alien, which I think is one of the deepest, most fundamental kind of human challenges we have, and how it's kind of played out in my work doing planetary science. But I wanted to begin by first saying just what an honor it is to be here. I told a couple people this at dinner. My father has terminal cancer, and when I was growing up, my sweet father, he would call me Ace, and it was such a sweet little nickname. And he's like, "Go get him, Ace," and just to be able to be invited here to be giving the Ace Vernon Lecture, and what an incredible explorer, somebody who really was contending with the truly alien, trying to find Sputnik, setting up Mount Cuba. I mean, it's just an extraordinary legacy, and I was so excited to meet his daughter, Lynn, who's here in the second row. And there she is, an extraordinary woman. For those of you that have not had a chance to meet her, you should meet her. And so I just wanted to say thanks again, and I think my dad is going to be very excited to hear about my Ace Vernon Lecture. Okay, so let's start off. Let's get right into it. So this, if you guys see this little red dot in the night, this point of light, this is Mars. This is what Mars looked like for so much of human history, looking out into the night and seeing this tiny little pale red lamp, sort of lighting up the sky. And for years, we go back to ancient history, and this idea of these wanderers, these planets that move separately from the fixed stars, they were really captivating the imagination. But Mars in particular was really enigmatic. This idea of the orbit of Mars, the path that it traced out in the sky. Every couple of years, Mars would sort of go eastward against the zodiac, but then every 26 months, for a few weeks, it would backpedal and go the different direction. And it was this retrograde motion that perplexed all these people. Plato even said, what must these be? Planets must have souls, because what else could these retrograde motions be if not acts of free will? So for a long time, Mars has been enigmatic. It has captivated us, and sort of time and again, we've had really different ideas of what this place was. And I wanted to start with Mars because Mars is the closest planet, of course, that we'll ever find, maybe, the closest planet that we know to Earth. It's the next planet over. Very similar histories, especially early on, around the time life was getting started here. But we've had so many twists and turns, so many blind alleys that we've been going down, and time and again, I think we've thought this is a world that we know, that we understand, when we don't have a lot of data, especially at the beginning, it's sort of this blank canvas that we can project a lot onto. And time and again, we've kind of seen ourselves, and we've also thought we've understood this world, only to find that it's really indescribably foreign. So I thought I'd give you a couple examples. So this one, our view of Mars, this was one of the first images from the Mariner mission, the Mariner 4 mission that went in 1965. It came back and it brought these images of the planet's surface, and you can see that this one was covered in craters. And it looked just like the pockmarked moon, and people were not expecting Mars to have all these craters, because that also means no plate tectonics, no liquid liquid ocean, no erosion, none of the sort of processes that we're so familiar with here on our Earth. But thank goodness we didn't just stop there. When we sent our first orbital mission, we realized that Mars had to have had a really different past because there were geologic features that could have only been carved by water, like this ancient river valley. So that was another big reverse course. When we went at the beginning with the Viking mission, these were the first landed missions on Mars. And I love this picture because it's got a blue sky. Turns out the sky does not look like that on Mars. They were doing some image processing, and this got released to the public, even though the sky on Mars is actually much more of a butterscotch color because it's filled with this dust that is so fine, it's almost the consistency of cigarette smoke. But this was released because I think the people that were processing the image sort of expected it to look like Earth. I mean, that sort of looks like the Arizona desert, right? You know, this is a place you can scrape your knee. Why wouldn't it have a blue sky? It was really interesting back then, too. We did the first life detection experiments. Here's old Carl Sagan with a model of the Viking lander out in the desert. And there were measurements there where we were looking for signs of life, and we didn't even find, this was supposed to be the greatest experiment in the history of modern science, and there were some really kind of confusing results at the beginning, but in the end it was sort of, it seemed that there were chemical processes that could have explained all of these things. But one thing that was really enigmatic were there were no organic molecules, the simple building blocks of life as we know it. None of those were found, and we should have had those, because even on the lifeless moon, we've got organics kind of raining down from space that are being delivered, and we couldn't find any on Mars. And it just seemed like it was the nail in the coffin for the possibility of finding life there. But, so here, this is showing the Viking arm, but fortunately we went back, here's 2008 with the Phoenix mission, which lands near in one of the high latitude regions of Mars, and we figured out what was happening. There was this salt that was laced throughout all the sediments there called perchlorate that was making it so the signal was just vanishing. The way we were trying to detect these organic molecules was heating them up and using a gas chromatograph mass spectrometer. And it turns out when you heat organic molecules in the presence of this incredibly corrosive salt, it's almost like you dry clean them and you get rid of the organic molecules so you can't even see them. Finally figured it out with this mission, which was really exciting. Again, another big reversal. And this is Opportunity. This was the first rover mission that I ever got to work on. Opportunity was roving around. This is this place called the Burns Cliff. You see these beautiful bedrock layers there. It turns out these rocks are filled with this mineral called jarosite, which is only formed in incredibly acidic conditions, like the pH of battery acid. And so then we had this idea, well, gosh, there's lots of water on Mars, but what if it's the kind of water that is very, very hard to survive in because it's so acidic, say pH 2. But we had this idea, well, gosh, what if Mars was just bathed in acid, all of its history? But then we go back. This is the Curiosity rover, one of the rovers I'm working on now. We found these beautiful mudstones as soon as we landed in 2012. And it turns out that these kinds of deposits had to be formed in the presence of what we think of as sweet water, water that's pH neutral. The PI of this mission got up at one of the landing press conferences and said, if you'd been standing there on Mars at this site a few billion years ago, you could have drank this water. And that actually Mars has this long history that preceded that time where the waters got acidic, where things might have been much, much more habitable. So lots and lots of backs and forths just looking at our next world over. And I put this is the parachute. You can see the most recent Mars mission is called the Perseverance rover that NASA sent in 2020. And this is collecting samples to bring back to Earth to look for signs of ancient biosignatures or traces of life. Incredibly exciting. I love that this orbiter managed to capture the landing parachute as it was going down in this place called Jezero Crater. It's just breathtakingly ambitious mission. I'm really excited about getting these samples back on Earth, but also really kind of begging the question, we are finally doing life detection. Again, we did that in the 1970s. The biggest experiment in the history of modern science failed. We didn't do any more life detection. We did a lot of, let's follow the water. Let's see if we can find signs of habitability. But now we are embracing that big, ambitious goal again of looking for signs of life. But it does beg the question of what is the best way to look for signs of life, to think about how we would go about detecting life. And this is the thing that's been really motivating to me in my career, like trying to understand, are we alone? Why is there something and not nothing? And did that something from nothing, did it happen once? Did it happen time and again? And these are the kind of questions that keep me up at night. And so what's the best way to look? We've got to learn how to look. We've got to learn how to better look for life as well. And one of the things that you could do is you can go out to places on Earth that have relevant similarities to other planetary environments, at times places that you think it's really hard to find life, places that were even thought to be sterile for years and years and years, where there are traces of life, but they're not necessarily easy to see. And you can learn how to look for biosignatures in planetary environments. And so that's one of the things that I do a lot of in my lab at Georgetown. We do things like exploring the limits of life. These are some basaltic lava tubes that are underneath the ground, looking in the sort of darkness underneath the Earth there, where microbes are surviving on very few nutrients and just chemical sources of energy, not light from the sun. I also do work trying to understand the persistence of life. There are a couple of field sites that I have that we have in our lab in Antarctica. These are the dry valleys, the 1% of Antarctica that is not covered by ice is a nice place to go and try to do research. This world was really different in the past, millions of years ago. There's been no rain in the dry valleys for 2 million years, but a long time ago, it was a much more warm and wet and clement environment. And they're ancient lakes, and they've left these beautiful deposits all the way up these shorelines in these valleys, because there used to be these enormous lakes that have since receded to these tiny little lenses of ice-covered lakes now. So we go and we look at how biomolecules are preserved in these dry, cold, polar deserts, these places that are very arid and dry, like Mars. This is another site here called Lake Untersee on the right that's just completely dominated by prokaryotes, where we don't have these predator-prey relationships, these very simple organisms. You can see all of these microbes that are forming these beautiful iridescent microbial mats, some of them up to 70 centimeters in height. And we think that much of the early Earth, like life would have looked like this, very simple. And so we do some, when you have structures like that, these kind of modern stromatolites, and how they get lithified over time. And we look for how you can find, fossilized molecules in these environments. And then part of what we do, too, is applying what we learn working here on Earth to space exploration, doing the kind of training and technique development to both analyze data sets and then also think about the next generation of tools and techniques that we'll want to use. This is Curiosity again. You can see the rim of Gale Crater there in the background. Here's Mount Sharp, this beautiful mountain. It rises as high as Mount Rainier above Seattle, contains this miles of sedimentary strata that record much of the climatic history on Mars. So we're kind of reading through, deciphering those records now. So as part of what we do, and this is not just me, of course, the whole astrobiology community is we've gotten really good at developing ways of detecting life as we know it. Like, we are really quite good at sort of going into these environments and finding life now and traces of life. So there are a lot of different ways we do this. A bunch of it is just kind of identifying established and widely accepted features that are associated with life and the signatures of life. We look for particular classes of molecules. We look for isotopic signatures. We go after patterns within the molecular weights of lipids and other lipids. We look at like fatty acids and other lipids. We look at biogenic structures and textures, biominerals. And we do this in extreme environments when we go out to places. These are some in Australia, these acid salt lakes we've been studying. The images on the left in the middle are images from Mars. The images on the right are images from Earth. Lots and lots of similarities in terms of the mineralogy and the features that we see in the geology. But we do it in extreme environments. We also can look back into the early rock record of Earth to find sort of the earliest traces of life. And we do it on other worlds. Here again is the Curiosity rover. The instrument that I'm part of is the Sample Analysis at Mars, or the SAM instrument. And it's looking at organic molecules, and here are some of the ones that we found. Again, those are the sort of building blocks of life as we know it. And there are all these benefits of looking for life as we know it. It's almost like if you lose your car keys and it's a dark night and you're out on the street, the first place you should probably look is underneath the street lamp, because that's where the light is and that's where it'll be easiest to find your car keys. And if they're not there, then you could venture out a little bit more into the darkness. But when you start from what you know, it's a lot easier to interpret the data. You have signals that can be really rich in information. And you also have a stronger understanding of this process of taphonomy, like what happens over time, the kind of diagenesis, where you start with something and it's not necessarily the same thing millions or billions of years later. It's kind of like if you are a geologist and you go off looking for a dinosaur, you don't expect to find the dinosaur. You'll find like a dinosaur bone. We can do the equivalent of that with molecules. You might not find the original biomolecule. Some functional groups might have fallen off, like fingers and toes, but sometimes you can still find the backbone of that molecule and you can reconstruct what it is. So we do a lot of that, and we've figured out sort of how to do that here on Earth. And again, as I was saying at the beginning, Mars is a great place to look for life as we don't know it, because Mars was so similar. It's really as similar as it gets to the Earth. You've got kind of similar size, similar original inventory of volatile, similar silicate crust. Once Mars, early in its history, was also protected by a magnetic field. It had a thicker atmosphere, one that would allow liquid water on the surface. Again, early on in histories, we didn't have this oxygen-rich atmosphere. Both the Earth and Mars had thick carbon dioxide atmospheres. And we know that Mars was at least episodically warm and wet in a much more sort of clement environment. So you're kind of starting with the same sorts of conditions, the same sort of starting materials. And then also, we know that Mars is really close by. And during, this period in early solar system formation, it's sort of debated how long the late heavy bombardment happened and how intense it was. But we know that billions of, tons of rocks were exchanged between these two worlds. And, I have colleagues who have gone in and cut into these samples, these meteorites that we've got from Mars that have landed here on Earth, and they've shown that the Curie temperatures are not above kind of sterilization temperatures. Modeling of some of the trajectories suggests that, some of these meteorites could have, you could have gotten them here from Earth, back and forth, Mars to Earth, Earth to Mars, on really short periods. And maybe there was a chance that life we just kind of caught it from the next planet over. Who knows? Maybe we're all Martians, right? There are more of those rocks coming in because of the gravitational well of the sun and going out. But we do have this possibility of exchange because, again, we are pretty close. And we do know that life can kind of take a beating and get through. So, all good reasons, all great reasons to kind of go after life as we know it as a starting point, but I think it's really important not for that to also be an ending point. And so I'm going to talk a little bit about the limits of analogy. And I really mean that in two ways when I say that. So there's also this idea of like understanding even what life is, you know, like we're really toddlers in a lot of ways. People will say like biology in many ways is a kind of just descriptive science. We have one data point. All the life we know of is the carbon-based, DNA-based life that's here on our planet. But as time has gone on, we've gotten a lot more sophisticated and even understanding that. And the example that I want to give is those Viking experiments that I talked about. There were 3 life detection experiments in addition to that GCMS that was looking for organic molecules. And one of those experiments, it was called the Vansoyama. It was nicknamed the chicken soup experiment. The idea was that you would take a sample, you'd scoop it up of the Martian soil, you put it into the instrument, and you'd have this nutrient-rich broth with the idea that you dump it in. These organisms on Mars would be just delighted. They'd be so happy. Every nutrient they could want would be in this really rich nutrient media, and they would grow and grow and grow, and we would see signs of metabolism, and we will detect life. And that was the premise of the experiment. But this was back in the 1970s, and we did not understand until the advent of genomic methods, when we started sequencing DNA and not requiring something to grow in a petri dish, that actually we had been missing the vast majority of the organisms on our planet. Like 99% of those organisms on our planet would not grow in a petri dish when we brought them back into the laboratory, and especially not on sort of timelines so we would detect them. But when we didn't require something to grow in front of our eyes in a visible way, once we started just being able to look at the genomic sequences of the organisms in that soil, we realized that there's just tons of stuff we were missing. And so I would say that if we were designing those Viking experiments today, we'd probably design them very differently based on that understanding of biology that has been progressing as time has passed. And so I mean it in that way, like what even is life and biology, but also just kind of these possibilities of life that are beyond the confines of our current imagination. And so this idea of life as we don't know it, this is kind of a really fun time to be thinking about this. This is something the National Academies has recognized. There was a big astrobiology science strategy report that came out recently. And one of their big recommendations was that the search for life beyond Earth, it really requires more sophisticated frameworks for considering the potential for non-Terran life or life that, doesn't have the same origin, and NASA should really support research into a novel and agnostic biosignatures. And so how wonderful that this is all percolating and this recommendation comes out right as some colleagues and I are putting in a big grant to NASA to do exactly this. So I'm going to get into some of the research we're doing to attack this question in just a second. But I really want to motivate it because we're not just looking at Mars for signs of life anymore. We're in this really exciting time where we've got new huge rockets like the SLS, the Space Launch System, rolling into being, where we are going to be able to reach destinations that are deep into the solar system, including the outer solar system, much quicker and be able to launch really big payloads. We've got new propulsion technologies like solar electric propulsion that are also, just opening up this whole new generation of missions. And, if only we had something like a tricorder for those Trekkie fans out there. I really do hope somebody found their keys. We would be able to just point it at somebody and say, what is this? Is this life? Is this not life? And so how do we kind of go about doing this these days? And I just want to show you, there are all these other worlds that we really are thinking seriously about. So This is back in 1979. This is before I was born, but this is this glimpse. This is the first glimpse of Europa, one of the large moons of Jupiter, from the passing Voyager 2 spacecraft. And it had traveled, like 500 million kilometers. It gets out there, and it gets this like weird image back where you see all these lines kind of crisscrossing the surface, and it was so weird. Like, are those ridges? Are those troughs? What could these things be? And it was really super confusing because this was supposed to be a moment of discovery, and then it was just like even more confusing after we saw the picture than before we saw the picture. And it's really interesting because it was around that same time, 1979, that there is the first paper that's coming out in GRL, Geophysical Research Letters, suggesting that perhaps there could be tidal heating because of the resonance with Io and Jupiter's orbit, where you get all of this friction that is generated from tidal heating that creates heat, even though you're so far from the sun, you're well beyond what we think about as that kind of traditional habitable zone. But there could be maybe liquid water under this ice. And so here we go, 10 years later, Galileo goes back, and even though it was having a lot of trouble with its antenna, it was still able to take these readings with their magnetometer that suggested really the best solution to this problem was to put in their model under a salty ocean, a brine ocean underneath this thick ice shell. And then you see much more detailed images too of all of these kind of pieces of ice that were moving around. And then of course now we have the surface of Europa. We know it's like a bloodshot eyeball there. We've got the Europa Clipper mission that's launching soon. That's going to go back and help us really constrain the nature of that ocean, which is going to be really amazing. But we think that there's more. So this is the amount, if we took all the water on all of our oceans, it's that much. And then we think maybe 2 to 3 times as much water is on Europa, this moon of Jupiter, underneath this thick ice shell. And you've got these lineae, this morphing and changing the surface. And we think that this water, this liquid water ocean, is global in nature, and that it's persisted as long as the Jovian system has persisted. So a long-lived global ocean. And you kind of think about all the possibilities, and then you might say, well, this is a world that's just totally bathed in radiation. Is life really going to make it there? But we have our favorite Deinococcus radiodurans. These are organisms that live in the cores of nuclear reactors that have figured out a way to persist despite the incredibly difficult conditions. So maybe, who knows? We also, this is some Hubble data suggesting that there are plumes coming out of Europa where we could be delivering samples of that underwater ocean, right out to space. How convenient if you want to send an inexpensive mission up there to sample. You don't have to melt through that incredibly thick layer of ice. You don't have to send a probe and figure out how it's going to communicate. You can just scoop some material up and go look at it, which is really, really fun. Another place that we also know has this continual kind of cryovolcano action where we're delivering brine from what we think is another global under ice ocean is this teeny beautiful moon of Saturn called Enceladus. So Enceladus is tiny, it's the size of England, but it's the brightest object in our solar system. It has this beautiful white snow from these cryovolcanoes, so all this liquid brine gets shot up and then falls back down on the surface, and the stuff that doesn't fall on the surface goes out and makes the E-ring of Saturn, which you can see quite easily. And this is another world where it's just really exciting to think about the kind of possibilities for potential life. It's probably one of these where we've got an ice shell, we've got an ocean, potentially with hydrothermal activity down here above a rocky core. Really exciting, and just another reminder of, you think these deep hydrothermal vents under so much pressure, so dark. You know, for a long time, we also thought the bottom of the ocean, those places here on Earth, would be sterile. But then when the first sort of lights of the first submersibles started cutting across the dark ocean floor, we see these 6-foot tube worms with these feathery red plumage, and it's just like, what in the world? But there's tons of chemical energy down there, and despite the incredible cold and the harsh pressure, you know, you get these warm fluids that are coming out of the oceanic crust. You get redox gradients where it turns out it's easy for microbes to make a living, and not just microbes, you know, you can create all kinds of complex ecosystems. You know, and I think this was another huge surprise that we didn't understand about the nature of life. We just thought the whole seafloor would be barren. Okay, and then another place, my last one, that I'm really excited about, especially when it comes to thinking about looking for life as we don't know it, is Titan, the biggest moon of Saturn. And so Titan's the only moon that we know of that has a thick atmosphere. It's a place with clouds and weather and pale hills and dark rivers, and there are lakes of ethane and methane. So it's a world with a solvent, but that solvent isn't water. It's, you know, it's this gasoline world where you've got all these hydrocarbons, and it's really fun to start imagining what the molecular and polymeric building blocks of life on a gasoline world might be like. We've been there with the Huygens probe. We landed, this is an image from 2005. We're going back with this incredibly cool mission called Dragonfly that's run out of the Applied Physics Laboratory at Johns Hopkins. It's a quadcopter. It's going to bounce around and take all these, you know, interesting measurements. It's going to be incredible. It's going to go in 27, and I think get there in 2034. So mark your calendars. It's going to be very exciting. And here's just a radar image showing some of those gorgeous lakes. Yeah, you can see those gorgeous lakes. This is a Cassini radar image. And again, like this incredibly cold world, minus 180 degrees Celsius, but you know, you get lakes all over. Okay, so this brings me to this big proposal that we were so serendipitous and lucky that we put together this team and submitted. So we've been calling it, and maybe we have some regrets about this, it's an acronym sort of within an acronym, Laboratory for Agnostic Biosignatures. So we call it LAB, you know, because it's like a laboratory. Yeah, so we've been calling it Laboratory for Agnostic Biosignatures because it's this big 5-year project that NASA has given us $7 million to do, and it's been really, the core question is really that question, like how do you contend with the truly alien? How can we search for life without presupposing, you know, specific biochemistry, a particular underlying molecular framework? How do we kind of open our minds to the different possibilities for looking for life, not life as we know it, but life as we don't know it. So this is, I love talking about this because I feel like it's a project that I'm kind of most excited about, like day to day, these days in the lab. And you might say, okay, that's super hard. That's kind of like trying to imagine a color that you've never seen. Like, how do you do that? And it is a super challenge, but it's also really, really fun to kind of think about the different ways we could go about doing it. And so I'm going to tell you a couple of the ways that we are trying to go about constraining this problem. Okay, so one of them is this idea of looking for chemical complexity. So as my colleague Lee Cronin on the team likes to say, if you found a 747 on the surface of Mars, you really might have no idea how it got there, but it probably wasn't random. And especially if you found like a whole fleet of 747s, like did they just get there randomly, just from different chemicals sort of, you know, bonding together? And we're trying to do sort of the molecular equivalent of that. Like if you find a really complex molecule, and especially if you find like a ton of really complex like copies of that molecule, you know, like, how is that like the 747? Or maybe it's like the Tesla Red Roadster in our day and age here on the surface of Mars. But would that be, is there a threshold above which it is just exceedingly unlikely or potentially impossible for such complex molecules in sufficient quantities to be the result of random processes? And so this is sort of nice because you don't have to presuppose, I mean, you could have life that's not even carbon-based, you know? Like, you can think about, like, different ways. And so when you think about complexity, you know, there are different ways to kind of go after it. You know, what is complex, you know? You can have big molecules with heavy molecular weights, but that aren't necessarily complex, that are just kind of polymers with repeating subunits where you just kind of make the same thing over and over and over and over again. But we've been working with a bunch of different things from mathematics, like graph theory, putting together these algorithms to explore chemical space. And we've been assigning this idea, this molecular assembly index. So basically, if you take any molecule and you break it down into all its pieces and then you put it back together in the simplest way, if that pathway is sufficiently long, because you're doing different things. So like you can see, you have to account for the simplifying feature of duplication. You sort of increase your molecular assembly index if you're just duplicating yourself over and over and over again. But you have to do sort of different things. And this is showing an example just with this particular molecule, ATP. So the idea is if you found something as complex as ATP on the surface of Mars or another world, and this is sort of showing like if you used a mass spectrometer, what the sort of data would look like, because mass spectrometer also breaks molecules down into little pieces and you get all the fragments and you reconstruct those. But using graph theory and starting to explore chemical space, it does appear that there's this threshold under these algorithms of around 15 where everything out here is made by biology or made by humans in a lab, essentially made by biology. And even the really complex organics that are out in space from carbonaceous chondrites that we've been looking at, those things are, and there are a lot of biological things too that are less than 10, but like, you know, once you find things that are above that, we can feel pretty confident. There does seem to be this kind of threshold where these things seem to be biological in origin. And so the idea that we could look for molecules that are really complex as a potential source of biosignature is really exciting, and especially because we can do it with mass spectrometers, and these are really high heritage spacecraft instruments. We send mass spectrometers to space all the time, and we know how to build them to withstand the space environment. And so here are a couple examples. Well, this is just one example, actually. It's this new instrument called MoMA, which is going to go on this rover, a European Space Agency rover called Rosalind Franklin. Love the name. It's going to go off to Oxyplanum, and it's going to land, and it's going to drill down into the surface and get samples and bring them up where they might be protected from radiation, and put them through this mass spectrometer. And it's really exciting because it does both gas chromatography, but it also does laser desorption, which will get around this perchlorate problem because we're not heating them up, we're making them in a plasma with our laser. So that is very, very exciting. And we can actually use these high heritage instruments that are already breaking molecules into little pieces to figure out, to estimate the molecular assembly index of the molecules that we find. And they don't necessarily, again, have to be like the molecules that we have here on Earth. And so lots of work and collaboration with the folks developing this instrument. There's an instrument very similar to this that's going to be on that Dragonfly mission. It's called DRAMS. It's going to go on Dragonfly in 2027, and so we've been working really closely with that team as well at NASA Goddard to figure out how we can use existing instrumentation, but kind of with this new approach to the data to try to estimate molecular complexity and see if we're getting things that are really, really complex in those samples, which is exciting. We have another way, and I'm going to try to make sure there's plenty of time for questions. We've got another way that we've been working at this problem, this idea, again, of complexity, but going at it in a different way. So one of the most exciting things that has happened in the world of biology in the last couple of decades, of course, is the genomic revolution, but we've also had rapid advances in miniaturization. So we get these teeny tiny little sequencers that can sequence DNA and RNA. And this is us. We're using one out in the Dry Valleys. It's this teeny little instrument. So much of the battle when it comes to sending stuff to space is you have to take some enormous instrument in the lab and shrink it down to something very small that can withstand the space environment and also is very lightweight and takes little power, and that's a hard challenge. So MoMA, this instrument that I was just showing you, that thing's about this big, and it started off as like a huge instrument that would take up a whole lab bench, and we've got it shrunk, which is really great. Turns out this company, Oxford Nanopore, has gone ahead and done so much of this work for us. These little sequencers are 10 centimeters long. They weigh 85 grams. They draw 1 watt of power. They are really changing the way we're understanding kind of inaccessible parts of our biosphere on Earth, because we can get this, we're developing systems for remotely telemeter data from places that it's actually hard to spend a lot of time. We're working on the sample processes, but I mean, hopefully we'll have things like this under the ocean and in Antarctica and places where there aren't a lot of scientists a lot of the time to help us understand kind of what's going on biologically. Okay, so this is exciting. You might say, okay, well, that's like the most not agnostic thing you could look for, right? Like this is looking for DNA and RNA. Those are very particular biomolecules associated with life here on Earth, and that is true. But we've been doing this fun thing, which I think has just been so much fun, trying to harness the power of sequencing to fingerprint the complexity of a surface. And so it turns out, like if you take, like we're using little pieces of DNA or RNA as reporters. So if you have single-stranded oligos or pieces of DNA or RNA, you know, you could say something 50 or 70 base pairs long, they'll naturally curl up and they'll have this kind of secondary and tertiary structure. And it turns out they'll bind to like all kinds of analytes, all kinds of ligands. So bear with me for a second. The idea that we're taking here, we're borrowing from this thing called SELEX, the systematic evolution of ligands by exponential enrichment. This is a big thing, like kind of in like the cancer biology world. You can take a bunch of these oligos, like millions of these randomly generated little pieces of DNA or RNA, and like out in the cancer world, like if you want to find, okay, you've got this particular type of cancer, you've got a breast cancer cell, say, you want to target it, you want to be able to find it in the body, you can take your huge library of millions of these little short-stranded oligos, you can incubate them with that target cell. A very, very tiny number of those oligos will actually bind in some way to that target cell, again, way less than 1%. But you can go through this process and get rid of kind of non-selective binding. And so there's this thing that's the fundamental basis of molecular biology. It's called the polymerase chain reaction. It's the idea that you can take a piece of DNA or RNA, and you can actually just amplify it up and have this enormous signal to noise. I mean, that's the way we're able to read DNA from like a single cell even, is we can amplify its genetic material until there's a big signal to noise and we can read it out with a sequencer. Okay, so, and this again is very targeted. This is like saying you know what you're looking for in advance. But what we're doing is we're trying to flip this upside down, saying that we don't know what we're looking for in advance. We just want to look at the patterns of binding. So first, let me try to convince you that actually a huge variety of molecules, this is also what we think is at the heart of what we call the RNA world, this idea of the origin of life here on Earth. We think that there were little pieces of nucleic acids that were sticking to clays like montmorillonite, and that was the original sort of templating surface when we started building up longer strands of nucleic acids. We know that oligos will bind, they'll bind to minerals like clays in the RNA world, they'll bind to peptides and proteins, to small organics, to metals, to mineral surfaces. They're all of these ideas. So the whole idea here, okay, let's imagine two kind of test cases. Imagine a simple cell, and even the simplest cell has a vast amount of information that's patterned on its surface, all kinds of places that things could bind. And then imagine you've got like a mineral grain with, here's like a crystalline network, a lattice here with like repeating binding sites. And the theory here is that if you've got all this interesting stuff on the surface of your cell, again, you start with a library of millions of oligos. very small proportion of those will bind, but you'll have more things and different looking things if you've got a complex surface versus fewer things and things that look more similar if you've got a less complex surface. And then of course you can go through this process of amplification and detection, and the idea is if you've got a complex surface, you get many diverse sequences detected, and if you've got a less complex surface, you get far fewer. And we can do really interesting things. Like we could do serial dilutions, for example, getting down to where we think that, right before the signal disappears, where you'd have like a single cell or a single mineral grain, and look at those patterns, because again, you get this huge signal to noise. And it's really exciting. So again, we are not looking for DNA or RNA. We're looking for like all kinds of things that we can use those little pieces of DNA and RNA as reporters to fingerprint the pattern of complexity. And so it's really fun because we are developing little sequencers anyway to send to space to look for life that shares a common ancestor with Earth, to look for signs of contamination, and now potentially also to look for life that has nothing to do with life on Earth except that you see a lot of complexity over a very small amount of space. Okay, so that's been fun. We've been working on this at Georgetown, also with colleagues at University of Texas, Andy Ellington and Eric Anslin. Super fun work. We've also, and I'll try to make sure I finish soon so we can get lots of questions, there's the idea of elemental and isotopic accumulation. We've got all these new tools, these machines like nanosims for biosignatures and SIMS instruments looking at isotopes and LA-ICP-MS. It probably sounds like world soup, but there's so many new kind of approaches for looking at elements in different ways that we could potentially see accumulation, like signs of chemical disequilibrium, something in a compartment that is separate from the environment in proportions that we wouldn't expect, and then using those to help target biosignatures. We also know that, we've seen life, it really will concentrate these scarce elements into discrete entities that are geochemically distinct. So even if you had a different biochemistry, if this process was still happening, and this is just showing, how incredibly enriched cells are beyond, when they're biotic cells, say beyond like the abiotic precipitation from seawater, you see things like manganese and iron and cobalt and nickel and copper and zinc, all kinds of enrichments that are really kind of anomalous and really interesting. And you can see those also at the macro scale. So there's this thing called a Redfield ratio, a really cool discovery. So the ratio of carbon to nitrogen to phosphorus in marine biomass in all the dissolved nutrient pools, it is almost exactly 106 to 16 to 1. And so you get this huge signal at the level of an entire planet of what's happening with biology, even at the bulk level of seawater, even after these microbes decay and decline. And so we've been doing some really fun modeling work trying to figure out, would we be able to identify a Redfield ratio in an exotic fluid? Would we see something like this that we might be able to pick out at a much bigger macro scale level? Okay, one more, and then I'll take some questions. I love this quote. So this wonderful physiologist once said, "Life is nothing but an electron looking for a place to rest." And so we've also been looking for signs of energy transfer. You can have microbes can utilize an active anode as an electron acceptor in much the same way that they would use an insoluble ferric oxide, for example, a ferrous oxide. So if you had your ferric oxide, you could use that as an electron acceptor during respiration. And what happens, and this is just showing a really simple setup, you see this marked and sustained increase in voltage. This was you can sort of see it every time iron sulfide tailings are added to this, the sterile control, you see a signal that just falls away. It just falls away, falls away, falls away. You get this abiotic oxidation, it happens quickly, it falls away. But you see this really sustained change in voltage, especially once the microbes figure out that this is an energy source, that time and again, you can And you can see it. And so we're setting up a bunch of experiments. These are colleagues at Harvard. Pete Gerges is one of the main collaborators there, with the goal of developing these kinds of in situ planetary probes where we could like stick something like this into, say, a surface water interface and see if there's redox chemistry there. We might even be able to like take different electrodes and put them at particular, like different potentials and figure out if there might be like different niches where organisms could be taking advantage of certain redox pairs as a source of energy, which is very exciting. But one thing I will say is, if you find a strand of DNA or a hopane, like there's certain molecules, these biomolecules on Earth that are just smoking guns, and you can say, oh, I know what that is. That looks sort of like life, but if I put it on a phylogenetic tree, it must have diverged from everything else we know billions of years ago. There is a lot of kind of definitiveness for some of these classes of biomolecules, and that's another strength of looking for life as we know it. But we might, if we're being more agnostic, be trading our definitiveness for inclusivity. So fortunately, it's not that we usually send one instrument out on a spacecraft. Usually we get to send 6 or 7. And so we've been thinking really carefully about like how we can use Bayesian methods to combine different approaches, different data sets to try to build up these kind of these thresholds, these likelihoods, trying to convert our measurements into likelihoods and thresholds, and really sort of trying to move from this binary, like, eureka, there's life. And that could happen, right? Maybe you put your probe in your Europa one day, you melt through the ice and a shark swims at you and you're like, whoa, it's life. But if it's more complicated than that, if it's just simple life, that like the kind of life that dominated our planet for the vast majority of its history, it might be more helpful to sort of think about the data in terms of like, well, this is what we would expect from abiotic processes alone, and here we're 3 sigma away from that, 3 standard deviations away from that. So we're also trying to push this idea of a conceptual spectrum of certainty. But again, I just wanted to sort of underscore, I'm going to leave with this one video, which I just like this video a lot, but it's just like, again, time and time again, like, it's just been so important to try to open our minds and to try to understand like just how alien things are. And contending with the truly alien, that is hard. And so even Mars, this place that again looks so incredibly familiar, has these beautiful, baffling things about it. And one of the things is the fact that the sunsets on Mars are this just incandescent blue color. And it's because of the optical physics. We got Rayleigh scattering that's dominant here on Earth. We have a much thinner atmosphere on Mars. This thing called Mie scattering actually is the predominant thing, scattering off the dust. And so we get this kind of opposite effect where instead of red sunsets, we get blue sunsets. And you can understand how that works, but every time you see this, it just totally takes your breath away. And I just thought I would play that to end the talk, and then we can take lots of questions. Okay, so here it is. See if it's gonna play. Kevin's playing the piano. Oh, okay. So I'll end it there, but I would love to take any questions. Anybody with questions, ask a question. Scott's got one.
Yeah, so the chicken soup experiment,
Yeah.
wasn't that positive? Didn't something happen there?
Yeah, well, so there was such a quick oxidation. So it's a labeled release experiment, is what it was officially called, where you get this really quick what we think is like, it really looks like a chemical oxidation. But it was a really confusing moment because this guy, Gil Levin, so he was the one that did the labeled release. He was like so excited, and he was getting cigars and champagne, and he was like, we have met our criteria, like we have seen this big reaction, like this is life. But then the fact that it had just fallen away so instantly, and also that it was such a big whopping response to begin with, it really started, and then when they repeated the experiment, it really started to look like that's just chemical oxidation, that is not biology. And so I think that was disappointing. But Gil will still say that, he thinks that life was detected. But yeah, it does seem like it was probably a more chemical oxidation event going on there. A great question, yeah.
Good evening, Dr. Johnson. My name is Tom.
Can you use the microphone?
How do you do? Come to me. Thank you for your presentation. It was great. And like you, I've been staring at the ceiling for the past couple of weeks since I knew you were going to come. And it's a very interesting subject, very intriguing, to say the least. One of the things that I'm going to use this prompter. So,
sure.
Okay. Given the idea that the laboratory is focused on detecting life as we don't know it, does the research also include methods for discerning artificial intelligence on exoplanets? Also, just to add to that, Dr. Seth Shostak at SETI is a big proponent of and advocating strategies for doing this, going after planets that receive a higher amount of solar energy and also are rich in silicon and trace materials. Would you like to comment on that at all?
Yeah, no, that's an excellent question. Okay. And I do have to say, I'm a bit of a homebody. I just do like our solar system, I stay right here at home. And the world of exoplanets is absolutely exploding. It's like one of the most exciting things. Thousands now of confirmed exoplanets, even many more thousands of potential exoplanets. We think that, most stars have planets, which is incredibly exciting. We haven't even detected an exomoon yet, but how many Titans and Enceladus and Europas are there out there waiting to be found? Incredibly exciting. Seth is extraordinary. Huge fan. He's out at SETI doing really great work. And it's been fun. There's this group, it's called Nexus, the Nexus for Exoplanet Science. And there are a lot of folks, there are a lot of methods. Again, we use really different methods. I'm much more of like an in situ kind of experimental person, and the exoplanet folks are remote sensing people. But even though they're different data sets, and they're maybe looking at spectra, and I'm looking at, like a chemical, output from a GC-MS or something like that, the ideas of how we approach the data and how we kind of classify what an anomaly is and sort of what we would go after in terms of biosignatures, and especially putting together multiple data sets, we found a lot of overlap there, which has been really fun. And so it's been interesting sort of thinking about all this stuff. And again, not really my turn again right here. I think I'm a restless sort of person, and I really want to find life and know that we found life. And exoplanets do seem like so far away, and the universe has a speed limit, and it's slow, and that's hard. But you never know. I think about Mars 100 years ago. The idea of going to Mars, actually sending sophisticated robots, bringing samples back from Mars that have been particularly selected to look for life. That was all science fiction. And, that's one human lifetime, right? And we've had the space age, and we've had this extraordinary revolution in our understanding of this world. And I think about, the closest exoplanets, they're light years away, but maybe these little tiny spacecraft that weigh a gram that we can power with lasers and that can relay signals back, a whole series of them. Who knows? Maybe we will actually be able to know these worlds just as intimately. They won't always just be aberrations of light, and we can discern some things about their atmospheres, but maybe we'll actually see them. And technology is changing so fast, and it's so exciting to think about not only the sort of next generation of space telescopes, but the new ways we might think about missions. Anyway, great question. And anybody that doesn't know Seth Shostak, I suggest you get to know him. He's amazing. One right here.
Hi, thank you for coming. Great talk. I was wondering, when you do find life as we don't know it, do you have a way to predict how it will evolve?
Oh, wow. Deep question. Yeah. Yeah. They're really interesting thoughts. Actually, it's kind of been like kind of off in the sort of realm of philosophy. Like I went to this really cool thing at the Library of Congress where they brought together all these people kind of thinking about different sort of ideas of life. And do you, like, is Darwinian evolution, which we think of as so central, the kind of how a lot of us define or think about life, like, is that required? You know, like, could there be like life without death? Could there be persistence? Could there be just kind of like information? Could there be life that's no longer like tied to chemistry, or at least the kind of chemistry we think of? I mean, it's just so fascinating to sort of think about. Like, could there be life without death? Could there? Maybe. Like, it's just been really kind of mind-blowing. So, like, how would it evolve? I mean, I just think we're such toddlers, as I said, when it comes to these sort of things because of the one data point. It's just like there's so many things that are out there for us to know and to figure out. And maybe there are like certain kinds of constraints. Maybe biology can only develop in certain ways or only evolve in certain ways. Like maybe there are different bottlenecks that are kind of universal. Maybe there aren't. And just really figuring out the sort of undergirding, you know, like we have a periodic table for chemistry, you know, like trying to really figure out, like, it's almost, there's another great philosopher out at University of Colorado, and she says, we can't even like talk about definitions of life. It's like trying to define water before we knew what hydrogen and oxygen were. You know, we could say, oh, it's wet, it's liquid, you know, it's kind of clear, but was ice water? Was purple water water? Like, we really did not know until we understood how that worked when we discovered, you know, hydrogen and oxygen, and they're bonded together, and what a hydrogen bond is, you know, and like all of those things. And maybe we're at that point with life. We just have to find some more data points. We have to have a better understanding of what it is before we can say really anything. But great question. I think there's another one right behind this gentleman.
Again, thank you for your talk. This morning, ESA website posted an article saying that they found carbon on Europa, and the most likely source of that carbon are the vents.
Oh.
So I was wondering if you were at all familiar with that information, or what do you, if you're not, what would your jump to conclusion be on that?
Oh, wow. So was it through, do you know what the data source was?
JWST.
Okay.
So they
From
took that. I didn't read the whole article yet.
Yeah.
But they posted it today.
Oh, how cool. Well, I have been in New York. My best friend has cancer, and so I have been with her all day today, and I came down on the train, so I haven't read that. I haven't seen it, but I can't wait to look. So that would be really, really exciting. And we do think there's hydrothermal activity on Europa. We think that there are vents there. And again, huge possibilities for redox chemistry, for possibilities for life. So I will have to go look at those observations. It's great. I've got a neighbor in Maryland who's actually the project manager for JWST. So whenever I'm like behind on my info, he can catch me up. So maybe tomorrow at the bus stop, if I get home, I can catch Mike and get those scoop. Scott's got another one, or maybe is this a student question here too? Maybe the student then, Scott.
First of all, thank you for the presentation. It was super interesting. I was wondering, you mentioned molecular complexity and like molecular complementarity as like a method for detecting like agnostic biosignatures. Are there like more minor ones for detecting life as we don't know it that you like didn't cover in the presentation that you know?
Oh, yeah. So there are some other things that we have been brainstorming about. The way these kind of NASA research portfolios work is you get like a certain amount of money and you have a certain amount of time. And so part of the reason that we focused on these four was, and so we really do, we have these kind of four themes, plus we have this kind of overarching computational theme that involves a lot of computer scientists from places like the Santa Fe Institute who are just so fun to hang out with. And so there are some other like ideas that have been sort of percolating around there, but we have really focused on these, I think also because they're a nice spectrum where we have really high heritage instrumentation with like the mass specs, and then we have things that are like really experimental. Like, I mean, the idea of like sticking a probe and looking for signs of energy transfer, like we don't really have anything that could do that right now. Like, I mean, we would need to really start from the beginning to like figure out exactly how you would do that and deploy that in the framework of like a space mission. But yeah, we have these fun brainstorming sessions and we come up with ideas, and like some of them actually we've got some students working on publications for, which is fun. But these are the main ones that we're focusing on.
Gotcha. Thank
But
you.
yeah, great question. Maybe Scott had another one?
Yeah, so, phosphine and Venus.
Yeah. Oh, lots of thoughts about phosphine. So I am definitely on the camp of what Carl Sagan once said, you know, extraordinary claims need extraordinary evidence. There have been a lot of questions about those phosphine detections. Part of it is like, where in the cloud deck were the detections even, and would that place be the same place we think that like the temperature and pressure conditions would be okay for life? There's been, at the beginning, it seemed like they were lining up. A reanalysis of the data seems like they don't line up very well. Phosphine is something we've been talking about, especially folks in the exoplanet community have been talking about. Like, I went to the Astrobiology Science Conference and heard awesome talks about how, you know, phosphine would be a great biosignature gas and we should be looking for it out in exoplanets, and then boom, here it is in Venus. And it was this kind of exciting thing. It definitely got a lot of fanfare. For those of you that weren't familiar with this discovery, it was this, you know, discovery of phosphine in the cloud decks in Venus. Phosphine is something on Earth that's produced by biology, and it's kind of hard to think about ways that it would be produced abiotically. Have we exhausted photochemical space? I do not think so. I mean, there are all kinds of lab studies, I think, that would need to be done to really convince ourselves you can't make phosphine in an abiotic way. I have friends who have applied for grants to do just that, and NASA has not been sort of funding that work up to this point, even though I think it's like a totally obvious, wonderful thing to do. So I am a little bit of a skeptic about that particular observation, but I think it's really exciting. We have been neglecting, I mean, I think the attention to Venus is incredibly exciting, and when we do get sort of something that's kind of a little weird, like we should totally follow up and see more. Do I think it's life? I don't, but if I had to guess, but I don't think that that like means the end of exploring and figuring out how it got there. And, you know, Venus is like the sad stepchild. We haven't sent missions to Venus in 30 years. We're finally, NASA's going to send 2 missions, Da Vinci Plus and Veritas, to Venus. And so all the Venus huggers, all the Venusian scientists among the planetary community are very happy. And, you know, obviously we have much better technology than we did decades ago, and it's going to be a real revolution. But we'll have lots more data soon. So let's see then. Yeah, 2 more here. TJ, do you want to do the student question and then another question? Yeah.
Oh, yep.
Thank you so much.
Oh, no, of course.
So, if life were to be discovered, what would you hope it would look like, by any chance?
Oh, beautiful. Iridescent purple. No. Oh my gosh. I mean, really anything. I mean, of course, you know, the idea of like, oh, if we could find intelligent life and we could talk to it or communicate. I mean, we can't even communicate with dolphins, but like, I mean, if we could find something, I mean, that would be the coolest, right? But even, I think a lot of people sort of think, oh, who cares about microbes? Like, I wash my hands. There's like a little microbial genesis every time I step on microbes. Who cares about microbes? You know, they're like, why would that matter? And I really do think no matter how you cut it, like even if you went on Mars and you found life, even if it was the same genesis of life but had been separated, it would almost be like replaying the tape of evolution, rerunning that great experiment under different conditions. I think that'd be incredible. But the real thing, the thing that I'm most excited about is that idea of a separate genesis. And again, I think it would just tell us so much, even the simplest little microbe, you know, because it would have gotten here in a totally different way. And I think it really does get at that kind of something from nothing and why, and especially if we found a separate genesis of life, like right here in our solar system, right in our backyard. I mean, it's just got to be like a big old hatchery of all kinds of different life forms out there. So I guess I'll say that. Another question?
Well, my name is George Wolkind. I'm the president of the Delaware Rock and Roll Society.
That's awesome.
Basically, we're compiling a history of rock and roll musicians that, you know, grew up in Delaware. about 30 years ago, I was also an investigator for the Mutual UFO Network. And I was involved in investigating contact cases of people who were having extraterrestrials, visiting them in their homes. I left the Mutual UFO Network after I got fired.
Oh no! What did you do to get fired?
I got fired because I think my extraterrestrials did not satisfy the conditions of their extraterrestrials.
Oh no! Did they not look like little green men? Is that
Well, my extraterrestrials happen to like Law and Order and television and Chinese food.
There it is. That sounds like some fun extraterrestrials.
So I was involved in a UFO case that took place in Clark Summit, Pennsylvania in 1992. I went up there not knowing anything, or expecting anything, but eventually what happened was my UFO partner, we were looking at this radio tower, and there was a light above the tower on Bald Mountain. And I said to Irene, I said, look at that light over there. And she said, oh, that looks like the Star of Bethlehem. And I said, well, the Star of Bethlehem is moving. Well, it came toward us, and to make a long story short, we got to meet Sergeant Calley of the Newton Ransom Township Police, and we shook hands, and I said, how long has been flying saucers flying over your town? And he said, oh, about 6 weeks. So we interviewed him and the families that were seeing these things. But the most dramatic thing that happened was Irene and I, that was my UFO partner, we were going down the road, this is like 1:30 in the morning, and we had been there 3 hours and seen UFOs flying for 3 hours. I saw this little light in the suburbs flying around, and I said, Irene, stop the car, stop the car. So I got out of the car, and I fell down 30 feet of sliding ice and snow, and I landed on the street, and I looked up, and 20 feet away from me was a flying saucer, 10 feet off the ground. And I was like, oh, they're real. I waved. Of course, my brother said, did you say the magic words to keep the death ray from hitting you? And I said, what was that? And it was, Gord Klaatu Barada Nikto. Well, I didn't know to say anything. I just waved and hoped the death ray didn't come out. But it went up in the air and kept flying and disappeared in the night. So to make a long story even longer, I realized that flying saucers and extraterrestrials visiting America was real. So my question to you is, do you think it's real?
Oh, wow. Yes, that sounds like an amazing story. I'll tell you, my mother thinks that we've been visited and that the reason that I have become a planetary scientist is because they want me for some reason. My father doesn't share that opinion. I think it would just be so amazing if we've been visited by extraterrestrials. And I've watched like a lot of science fiction too. I've been watching Star Trek with my kids recently, which has been super, super fun. I love the idea of like intelligent life out there. And I think somewhere, like out in this enormous universe with all of the different galaxies, there's got to be something. I haven't had the experiences that you've had, clearly, but when I think about the sort of lines of evidence and the sources of evidence, I haven't seen anything that's felt like a smoking gun to me.
Well, thank you. I enjoyed your lecture. I learned a lot. I mean, this was really great.
Oh.
But the next time I see them, I'll give them your address.
Okay, perfect. Yes, my mother will be and they're vindicated. Maybe another student question here?
Former student, but close enough.
Okay, former student. I know, when I still sometimes occasionally get mistaken for a student, I take it as such a compliment. It's like the nicest thing anyone can say now that I'm an old lady.
As long as you don't say it, you're in the clear. So you talked about mostly samples that require physical contact as your area of expertise with sequencers, GC, and mass specs. But what are the methods for remote sensing and their advantages, disadvantages, and limitations in comparison?
Yeah, that's a great question. So you have an extraordinary range with remote sensing. And again, this world of exoplanets just massively exploding. It's been really exciting because probably when I was your age, it was back in the 90s when none of this has happened. And then it's just incredible. It's just been incredible. And of course, with remote sensing, you can look so much further in so many different places, all kinds of different worlds. And so I think that that's a real advantage. Even here at home, you can only do kind of targeted analyses and, very finite resources, like very limited number of places. And even Mars, we know Mars so intimately at this point. But Mars is a world that's every bit as enigmatic and complex as the Earth, and where do you go? And imagine setting down a spacecraft in the Antarctic Dry Valleys or the Amazon rainforest. You get really different results, right? And one place would be really easy to find, and one place would be really hard. And so I think targeting is really key, and that's why it's been great to have really comprehensive remote sensing data of a lot of these planetary targets. So a lot of the times we have reconnaissance missions, so we'll do a lot of mapping, and then we find things like Opportunity, the first rover that I worked on, that mission to Mars, it went because we did all this remote sensing work, and there was this mineralogical signal that just stood out like a beacon. There was a hematite signal, and hematite can form in the presence of water, and it was so weird that we just sent a mission there because we didn't understand it. Maybe there's some water there, it was a really weird thing. And then when we got down on the surface, that hematite was in the form of all these little ball bearings. It looked like Fred Flintstone with slip or something. There were all these perfectly spherical concretions made of hematite littered all over the surface, and it was almost comical. What? It was so surprising. And we called them blueberries because they had this kind of little blue tinge to them. But I think that the remote sensing is just incredibly helpful in targeting things. I just don't like to stop at remote sensing. I like to keep going, and actually get your hands dirty. I don't know. But again, I'm a restless sort of person. Yeah, great question. There's another one back here.
Thank you very much. I'm Jay Bancroft. I wonder if you have any advice for, there's such, everybody loves to work for NASA. You really have an enthusiasm for life and an appreciation perspective that maybe I was thinking for a layperson or a small-time politician might be trying to inspire people about the environment that is really kind of has a sort of a dark overtones when you work on it here. And do you have any sort of inspiration for how we take better care of our own planet?
Oh.
Thinking about your own studies?
Yeah, no, I think that's a wonderful, wonderful question. And I do a lot of teaching. I really do a lot of teaching of environmental science at Georgetown. I think that almost probably many of you guys that care about astronomy and care about planets, care a lot about this place too. There's no planet 2.0, we're never gonna make Mars just like Earth, and it's never ever gonna replace kind of what we're doing at a really massive scale in terms of pollution and climate change and all the things here. And I feel like I just have this reverence for life on Earth that comes from the studies that I've done. I feel like, especially thinking about things kind of in geologic time and these big vast scales and imagining, we've also taken pictures of Earth, Cassini turned back and through the rings of Saturn, sees this little pale blue dot, and it's just so precious and so extraordinary that we're here, that we exist, and that we have this beautifully, perfectly tuned world for us to live in. And it's so short, all these things are going to just collapse, and we're here for the briefest of moments, and we just need to take care of this world and all appreciate it as well, because there's just nothing else like it, like in the vast abyss out there, it's just this. But I think about that a lot. Like I ended up, I took a couple of years out of my postdoc to go kind of work on some environmental issues in the White House, because I felt like this kind of urgency around them. But I love the chance to kind of combine exploration and kind of asking these big questions, but with also working with students. And I do really like teaching kind of environmental classes to kind of help students, and like the students here, really take charge, because this is going to be the next generation, and we're mucking up the world, so you can help us take care of it. Yes, Dermot.
I wonder if you would comment on the current status of methane in Mars, especially the seasonal eruptions?
Yes, so interesting. So this is one of the big controversies on Mars right now. What in the world is going on with the methane? So this is something that the SAM instrument on the Curiosity rover has measured. There have been telescopic observations, dating back to, I think 2003 was the first one, of methane on Mars. And there's another instrument, a European orbiter, that's got an instrument it's called Trace Gas Orbiter, and it's got instruments measuring methane. And so we have really interesting results. So there's definitely some sort of methane on Mars. We see it on the surface with Curiosity. We see a seasonal pattern. We also see interesting diurnal patterns with the methane. Strangely, they do not match up to what the Trace Gas Orbiter is seeing at the same time in the same place. So there's this idea that there's this probably diffusion happening where we're not able to see the same sort of signals at orbit, but there might be a small inversion layer close to the surface. And this is a big mystery, because there are different ways to create methane. 90% of it here on Earth is biogenic. Created by microbes. There are ways through serpentization, other geologic processes, where you can create methane. But the sort of conditions that we understand about Mars, like Mars is more geologically active than we really understand if it's creating this methane, or there's a biogenic source. So it's a really exciting, super exciting sort of thing. There are some great researchers that are on our SAM team that are, helping to develop the sort of models, that there could also be like kind of ancient methane and clathrates, kind of little ice cages that are melting, that are being released up into the atmosphere. But I think the best source right now, and again, like this is an active, ongoing mystery with tons of people working on it and thinking about it, but I think that some process potentially akin to some serpentization in the subsurface, and then I think we're seeing a buildup during the night, and then we see levels that go down during the day, and I think that's because of the thermal dynamics, and then there is this kind of inversion layer that gets set up in certain parts of the year. But we do see like a pretty robust seasonal signal now since 2012 when we landed. So something cool is going on with the methane. Exactly what is still TBD. Sorry. Oh, they're gonna kick us out.
Shut down for the night, so I want to say thank you.
Oh, thank you. Thank you.